In this lesson, we will be discussing the mechanism of cellular respiration. In the previous lesson, we have already discussed the types of cellular respiration, that is aerobic respiration and anaerobic respiration. But in this topic, the SLO is explain the mechanism of cellular respiration. So we are going to discuss the all the steps which are involved in cellular respiration. So you already know that cellular respiration is series of metabolic reactions or metabolic processes that take place within a cell in which biochemical energy is produced from organic substance, that is glucose, and stored in the form of ATP to be used in energy requiring processes of the cell. Cellular respiration is an oxidation reduction reaction, or together they are known as redox reaction, because you already know what is oxidation and what is reduction. Oxidation is actually gain of oxygen or loss of electron or hydrogen, and reduction is gain of electron or gain of hydrogen. So what is happening in this reaction? Glucose, which is an organic substance, it undergoes oxidation, or it combines with oxygen, or we simply say that breakdown of glucose in presence of oxygen into its components, which are water, carbon dioxide, and energy is released. But cellular respiration is not as simple as this reaction. Rather, it undergoes series of metabolic reactions. We will discuss one by one. So it is oxidation reduction reaction, that oxidation of glucose is being oxidized into carbon dioxide, because loss of hydrogen or removal of electron is actually oxidation. And reduction of oxygen, because oxygen gains hydrogen molecules. If it is gaining hydrogen, it means it also gains electrons. So gain of electron is oxygen is reduced and glucose is oxidized. Moving on. So this is a brief summarization of cellular respiration. Cellular respiration is divided into following steps. First one is glycolysis. Second is oxidation of pyruvate. Third, the Krebs cycle. And fourth, electron transport chain. In this diagram, you can see that glycolysis is the first step of cellular respiration, and glycolysis is a common step both in aerobic respiration as well as anaerobic respiration. Aerobic respiration, if oxygen is available, we already discussed that if oxygen is available, then breakdown of glucose is complete oxidation of food occurs in cellular respiration, and this pathway is actually representing aerobic respiration, and this pathway is actually representing anaerobic respiration. So glycolysis is breakdown of glucose into pyruvate molecules, which is a common pathway, common step in both anaerobic and aerobic respiration. The second step is for aerobic respiration, because in anaerobic respiration the glucose is broken down into pyruvate, and then pyruvate is broken down either into alcohol or lactic acid. That is the types of anaerobic respiration. If it is broken down into alcohol, it is alcoholic fermentation. If it is broken down into lactic acid, that is the lactic acid fermentation. We already have discussed this topic in the previous lesson. Here you need to understand the second step is for aerobic respiration. After glycolysis, the pyruvate molecule has to enter Krebs cycle, but it cannot participate in Krebs cycle directly. That's why this glucose, before this pyruvate, before entering the Krebs cycle, it undergo oxidation and it is converted into acetyl CoA, and then it participates in the Krebs cycle. We will discuss it in the later slides. Then the Krebs cycle, it is the energy rich substances are produced in the Krebs cycle. So those energy rich substances like NADH and FADH, they are converted into ATP in the electron transport chain. Let's discuss these steps one by one. Here is again the overview of cellular respiration. First step is glycolysis, that is glucose is broken down into two pyruvic acid molecules which are also known as pyruvate molecules. Glucose is a 6 carbon containing compound and pyruvate is a 3 carbon containing compound. So one 6 carbon containing compound, when it splits, it will form two 3 carbon containing compounds, and as a result ATP and NADH are produced in glycolysis. What happens to these pyruvic acid molecules is the next step. These pyruvic acid molecules, in presence of enzyme coenzyme A, they are converted into a 2 carbon containing compound, that is acetyl CoA. This acetyl CoA participate in the Krebs cycle, and during that carbon dioxide is released. ATP, NADH and FADH high energy molecules are synthesized, and the third step, in the last step of the cellular respiration, these energy rich substances move to electron transport chain, and in electron transport chain they are utilized to form ATP. In electron transport chain, one NADH gives 3 ATP molecules and one FADH2 gives 2 ATP molecules. It gives 3 ATP molecules and it gives 2 ATP molecules. Let's discuss glycolysis first. So this is the first step of cellular respiration. I already told you that glycolysis is the process in which one glucose molecule, that is 6 carbon containing compound, it is broken down into two molecules of pyruvic acid, also known as pyruvate. The glycolysis is a multi-step metabolic pathway. There are so many steps, but at your level you can skip the steps, and you just need to understand that glycolysis is that process which takes place in the cytoplasm. This glycolysis actually occurs in the cytoplasm, and the next step, that is Krebs cycle and electron transport chain, they take place in the mitochondria. That's why mitochondria is known as the powerhouse of the cell, because the actual energy ATP that is produced in the mitochondria. We will discuss it later. First, let's see what is happening in glycolysis. One glucose molecule is going to form two pyruvate molecules. The end product of glycolysis is two pyruvate molecules. So these are the steps, and it is not necessary that you have to remember each step, but it is enough that you just need to understand that glycolysis has two major processes or two major phases. One is energy investment phase. You can differentiate them from the color, like there is a difference in color. The above darker color represents energy investment phase, and this color actually represents energy payoff phase. Energy investment, here what is actually happening, glucose molecule when enters the cytoplasm of a cell, it is converted into, there are series of reactions, but I have chosen this diagram because it is very direct and it is very simple to understand at your level. So what's happening, glucose when it enters the cytoplasm, it utilizes energy. Two ATP molecules are invested. This is energy investment phase, because here energy, instead of producing energy, in order to be broken down, this glucose first actually invests energy. It uses ATP molecules to convert this glucose. First it will use one ATP and it will be converted into fructose 6-phosphate, then it will utilize another ATP and it is converted into fructose 1,6-bisphosphate. So this is again, fructose is a isomer of this glucose, but the difference is that it has two phosphate groups. Now it is charged and it cannot leave the cytoplasm. Once the glucose is entered in the cytoplasm, it is converted into a polar substance, that is fructose 1,6-bisphosphate. One phosphate is attached at carbon number 1 and another at carbon number 6. That's why it is known as glucose, fructose 1,6-bisphosphate. And soon again this fructose 1,6-phosphate, it splits into 2,3 carbon containing compounds, which is actually going to be converted into glyceraldehyde 3-phosphate. In photosynthesis we also discussed this, you are familiar with this name, G3P. This is a 3 carbon containing carbohydrate. So this there we have skipped many steps, so there are actually 10 steps, and each step of the glycolysis need different enzyme. So glycolysis is a enzymatical process because it needs certain enzymes to complete the processes. So this 3 carbon containing compound, each of the 3 carbon containing compound undergo same changes. They undergo same changes, and it is going to gain it is going to release phosphate group, and then it is going to produce NADH and 2 ATP molecules. Same is happening on this side. 1 NADH molecule and 2 ATP molecules are produced, and the end product of glycolysis is 2 pyruvate molecules or pyruvic acid molecules. So here you can see this is payoff phase. Here 2 ATP molecules were used, and here 2 for this site and 2 for this molecule. So 4 ATP molecules are synthesized. For net energy gain of glycolysis is 4 ATP molecules. 2 were used in the investment phase, so we call net ATP production of glycolysis is only 2 ATP molecules and 2 NADH molecules. We will discuss the energy from the ADH molecule in the electron transport chain. Moving on, this is oxidation of pyruvate. So what happens once the glucose is broken down into 2 pyruvate molecules in the cytoplasm? Now this pyruvate molecule has to participate in the Krebs cycle, and Krebs cycle actually takes place in the mitochondrion, and this pyruvate is actually present in the cytoplasm. So it has to move to the mitochondria. It has to cross the mitochondrial membrane, and it will reach the mitochondrial matrix. So what happens actually, before entering the Krebs cycle, each pyruvate molecule, there are 2 pyruvate molecules, each of them is converted into acetyl-CoA, which is a 2 carbon containing compound in presence of coenzyme A, making 1 NADH molecule. So here you can see yield is a 2 carbon containing compound with the release of carbon dioxide and 1 NADH molecule. This NADH molecule is going to yield ATP in the again electron transport chain. So this was about pyruvate oxidation, that pyruvate cannot participate directly in the Krebs cycle, so it undergoes series of changes and it is converted into a 2 carbon containing compound in presence of enzyme coenzyme A, producing acetyl-CoA and releasing 1 NADH molecule. Moving on, Krebs cycle. So Krebs cycle involves series of enzyme catalyzed reactions that reduce the acetyl-CoA, which was formed from the oxidation of pyruvic acid, right? Pyruvic acid oxidation, it is oxidized and it forms 1 NADH molecule and it releases 1 carbon dioxide. Now this acetyl-CoA, in presence of this coenzyme A, this acetyl-CoA can participate in the Krebs cycle, and there are series of chemical substances. The first product of Krebs cycle is citric acid. That's why it is also known as citric acid cycle. So again, you don't need to remember these steps. You just need to understand that the acetyl-CoA combines with 4 carbon containing compound, and it forms a 6 carbon containing compound. It undergoes series of steps, and as a result, the output of Krebs cycle is formation of 1 ATP molecule, 1 FADH molecule, and 3 NADH molecules. NADH you already know. NAD plus is a coenzyme, and nicotine amide adenine dinucleotide is a coenzyme. When it reduces, it forms high energy molecule, and it is going to form ATP molecules in the electron transport chain. Same is the case with FADH. Flavin adenine dinucleotide, FAD, is also a coenzyme, and it gets two hydrogen ions and reduces to FADH2, which is a energy rich substance. So the output of Krebs cycle is one ATP molecule. One Krebs cycle we are talking, because for one pyruvic acid there is one Krebs cycle, and for two pyruvic acid, because resultant or the output of glycolysis was two pyruvate molecules, so each of them undergo this Krebs, it will enter the Krebs cycle, and each the pyruvic acid is going to form one ATP, I am repeating it, one FADH2 and 3 NADH molecules. Moving on to the last step, electron transport chain. So the energy rich substances which were formed during glycolysis and during Krebs cycle and during the oxidation of pyruvate, they are converted into ATP during the electron transport chain. What is electron transport chain? It is a series of complexes. They have been represented through stairs. Actually, they are series of protein molecules which act as electron acceptors and electron donors. So the electrons from the high energy molecules, they are passed to these molecules, and when these electrons move from one molecule to the next, their energy is used to make ATP. This is the form of energy which the cells can directly use. See, electrons from electron donors to electron acceptors by redox, both oxidation and reduction occurring simultaneously, and couples this electron transport with transfer of protons across the membrane. Oxygen is the final acceptor of the electrons. So what is happening actually during the electron transport chain? The electrons are moved from one electron acceptor to the next. So when these electrons are moved, due to the movement of these electrons, hydrogen ions are pumped out through a protein which is actually ATP synthase enzyme that pumps the hydrogen ions, and that pumping of hydrogen ions, the energy from those hydrogen ions is used to make ATP. So where these electrons are going, oxygen is the final acceptor of those electrons, and it results in formation of water. So here you can see in electron transport chain, one NADH molecule is going to make 3 ATP molecules, and one FADH2 molecule is going to form 2 ATP molecules. See, here you can see this is a overall general summary of the net ATP yield of cellular respiration. All the steps have been mentioned here. See, glucose is broken down into pyruvate molecules in the first step, that is glycolysis, and in glycolysis 2 ATP molecules were synthesized. Net ATP production of glycolysis is 2, because 4 ATP were synthesized but 2 were utilized in the energy investment phase, so we call that net ATP production of glycolysis is 2. And the NADH molecules in the glycolysis, there were 2 NADH molecules, right? So 2 NADH molecules, I already mentioned in the previous lesson, the previous slide, that one ADH molecule is equivalent to 3 ATP molecules. So here 2 NADH molecules were supposed to give 6 ATP molecules, but in glycolysis it is giving 4. The reason is, I have mentioned here, the NADH gives 2 ATP because 1 ATP is spent to transport it across the mitochondrial membrane. This is talking about glycolysis. In glycolysis, the NADH gives 2 ATPs because 1 ATP is spent to transport it across mitochondrial membrane. Of course, glycolysis took place in cytoplasm, and in order to transfer this pyruvate molecule into across the mitochondrial membrane, 1 ATP is spent. That's why we call that NADH of glycolysis gives only 2 ATP molecules in eukaryotes, but in prokaryotes Krebs cycle also occurs in cytoplasm. That's why net ATP yield in prokaryote is 38 ATPs in prokaryotes. Due to this minor difference, like there they were supposed to be 6 but they are 4. Due to this difference, net ATP yield in eukaryote is 36 and in prokaryotes it is 38. So we are done with this portion. Now I already mentioned that the next step was that the pyruvate undergoes oxidation and it forms acetyl-CoA. During that, each pyruvate makes 1 NADH. So for both pyruvate molecules, there are 2 NADH molecule, and 2 NADH molecule are going to make 6 ATP molecules in the electron transport chain. And now once the acetyl-CoA enters Krebs cycle, acetyl-CoA from each pyruvate is going to enter into the Krebs cycle, and in Krebs cycle, one Krebs cycle we discussed that 1 ATP, 3 NADH and 1 FADH. But for both the Krebs cycles of both the pyruvate molecules, ATP they will be double. 2 ATP molecules, direct ATP to 6 NADH, 6 3s are 18 ATPs, and 2 FADH molecules, 2 2s are 4. So when we sum up all these ATP molecules, the net ATP yield of cellular respiration or aerobic respiration in eukaryotes is 36 ATP molecules. In prokaryotes, I repeat it, it's 38, because in prokaryotes glycolysis, oxidation of pyruvate, Krebs cycle, electron transport chain, they all take place in the cytoplasm. However, in eukaryotes, glycolysis takes place in the cytoplasm and the rest of the processes take place in the mitochondria. Here is the summary. See, this is cytoplasm and this is mitochondrion. In cytoplasm, glucose undergoes glycolysis and it yields 2 ATP energy input to start, and 2 ATP is a net, I already discussed in the above slides, and it produces 2 NADH, and these NADH they all the high energy molecules from the glycolysis, from the oxidation of pyruvate, from the Krebs cycle, they all undergo they have to travel to the electron transport chain, which is series of proteins that are located in the mitochondrial membrane. They transfer the electrons through different electron acceptors, and finally, this is an important step, you have to remember that finally the electrons are accepted by oxygen and they combine with a hydrogen ion and they yield water. The water which we mentioned in the reaction of cellular respiration, that water is released during electron transport chain, keep in your mind. So the total ATP molecules of electron transport chain are 32. In Krebs cycle, direct ATP 2, in glycolysis 2, 32, 34 and 36. So again the net energy yield of aerobic respiration in eukaryotes is net yield is 36 ATP molecules. So this was all about today's lesson. Hope it was clear to all of you. If you have any questions, you can ask me in Google class, or you can approach me in WhatsApp group as well. Thank you for listening, class.